And I would like to invite the next speaker, Sebastian KVIST. I hope he's with us.
I'm
Sebastian?
here. I'm here, Sergey.
So it's your turn now. Your report. Mm-hmm.
Perfect. Let's see. Can everyone see my screen right now?
Yes, we can see.
Okay, very good. Thank you, Sergey, and thank you to Dr. Ceylon for inviting me to this international symposium. It's quite an impressive setup of speakers and an impressive setup of topics, and I see a lot of people have tuned in, so I'm very happy to be part of it. I apologize for any lighting issues. It's quite early on in Toronto, Canada, where I am, so it's early in the morning, and I'm trying to give you some light at least so you can see me, because it's very dark outside still. Okay, so thank you, Dr. Saglam, for a neat segue into biotrafficking and into contraband leeches, which is what I'm going to be talking about. There's I'll talk quite specifically about a certain project that we undertook in 2018-19, and it was published in 2020. So this is a very specific project, but I'd like to talk more generally if anyone has any questions later on about contraband leeches and in particular how they are brought into Canada, I would say. Okay, so before I get going, I'd like to say that this is a collaboration between a lot of different people, and so I'm not, I'm the lucky one that gets to present this work, but it's a collaboration between both Canada and North America. And Kalani, Megan, Yvonne, Mai, and Mark were instrumental in putting this down on paper, of course. So I wanted to start off with mentioning the collaborators. Okay, so I thought that I might give you a background into two different things. First of all, the concept of iDNA, which might be new to some people. iDNA technically stands for invertebrate ingested DNA. Essentially, it's the study of the DNA inside of blood-feeding organisms. And for leeches, of course, this means the DNA inside of the blood meals in the gut region. The concept of iDNA has been used to monitor mainly mammal diversity in certain regions of the world. And so basically what it is you get the DNA out of the leeches, out of the blood inside of the leeches, thereby preventing the need to collect the mammals or catch the mammals or potentially harming the mammals. And so you actually get DNA from the mammals through the ingesta of the leeches. But of course, blood meals inside of leeches hold a lot more information than simply what animals are present in an area. Most importantly, I think for myself and my group, my lab here in Canada, is that we don't really understand the true diet of leeches. We're not really sure what leeches actually feed on. We have found that some leeches that we believe are strictly blood feeding actually do feed on lots of different food stuffs, and not only blood. We've seen that they have ingested fish larvae. We see that they ingest frogs' eggs, etc. And so, iDNA can allow us to understand the broader ecology of leeches, as well as monitor mammal diversity, bird diversity, reptile diversity in certain areas. So that's basically what ingested DNA has been used for previously. It's been used, the first paper I'd say that was quite comprehensive came out in 2012, where a bunch of leeches were studied in Vietnam. And these are terrestrial leeches, and I'll show you a couple of photos of the terrestrial leeches later. It's the family Hemidipsidae, which is quite different from the family Hirudinidae, but they're, of course, both Hirudiniform, so they're part of the same suborder, but not part of the same family. But Schnell and his colleagues in 2012, they focused a study on trying to understand the mammal diversity in Vietnam by collecting several leech species and sequencing the blood inside of the leeches. And they found 26 leeches. 21 of them actually had blood or had DNA inside of their guts that they could amplify and sequence. And they found a variety of mammal DNA inside of the blood. In fact, some of the mammals were thought to be extinct, Some of the mammals were critically endangered and hadn't been seen for quite a while. And so this study actually allowed researchers to understand that leeches could be used to monitor mammal diversity, and specifically when it comes to mammals that are otherwise difficult to find in camera traps, etc. Could be used, they could be found through the use of iDNA in leeches. Moving along, another study that used iDNA in leeches as well, Maifami and colleagues used iDNA from hemidipsid leeches in Madagascar. And this chart shows the different taxa that they found, whose DNA they found inside of these leeches. And in green, you'll see mammals. In yellow, you'll see amphibians and reptiles. Sorry, in red, you'll see amphibians and reptiles. And in blue, you'll see birds. And so this chart is just simply to show that there's a wide diversity of prey stuff for these different leeches. Now, these are hemidipsid leeches. We know that they're a bit more aggressive than their freshwater counterparts, perhaps. And by aggressive, I mean that they have a wider array of hosts. And so we think that they feed on a wider distribution of hosts, and that's why we're finding so many in this study. But I wanted to show you this chart simply to show the amount of prey items that these leeches have. Okay, so basically through the study of iDNA, we can monitor the distribution, the geographic distribution of mammals, of birds, of amphibians and reptiles across the world, but also gives us an understanding of the diets of leeches, which can help us understand the ecology and the behavior of certain leeches. But it's also the case that leeches have been used, for other purposes than simply to understand the distribution of animals across the world. There was an instance of a leech in Australia where there had been a burglary, and the police got there and they found no evidence or no traces of any person, any burglar there. But what they did find was an engorged leech on the carpet, and so they took the leech and they sequenced the DNA inside of the leech, and they didn't get a match in their database. But 8 years later, a person was pulled over for speeding, and he was checked for his DNA was checked, and lo and behold, they got a match with this burglary 8 years before. So this leech actually helped crack a case of burglary in Australia, and that's all due to the ingested blood that's inside of the leech, and the fact that the Australian police actually were forthcoming enough to sequence the DNA right at off the bat when they found the leech, instead of waiting the 8 years. We think the blood inside of leeches actually stays, the DNA stays intact for at least 6 months, but probably up to close to a year. That's what we're finding in empirical studies. Okay, so what I've been discussing right now is hemidipsid leeches, right? These are mainly terrestrial leeches in Southeast Asian tropical forests mainly. And the question that we had is of course, can we use iDNA in freshwater species? Can we use it in marine species to try to estimate the diversity of prey that they feed on, but also the distribution of the different prey stuffs around the world? And so hemidipsids have been quite heavily studied for the purpose of iDNA, but a lot of other taxa still need to be studied for this purpose. Okay, so that's the background for iDNA, just to get everyone to the same level, because it will be important in this story as we move forward. Okay, so this is the study basically. In 2018, I was contacted by the Canadian Border Protection. And Wildlife Officer Mark McIntyre called me up and said that we have confiscated 5,000 leeches in customs. And they're on their way to Canada from Russia. And they were brought on the plane in plastic bags, and somehow there was water in the plastic bags. And we're not really sure how he got to bring liquid onto the plane, but he managed to keep these leeches alive. So the question that he had was, can you identify the species for us? And I said, yes, absolutely. And he brought us some 500 leeches in a bag to the Royal Ontario Museum where I work in Toronto, Canada. And we were quite quickly managed to identify the species. Of course, Hirudo verbana, we certainly compared it to the other known species of Hirudo. But we figured out quite easily that it was Hirudo verbana simply from the dorsal color pattern. But that was also later confirmed by a genetic analysis, of course. Okay, so these 5,000 leeches were confiscated in customs. This made quite a splash in Canadian news, at least, and across the world, where people hadn't really understood up until this point that the leeches were used in biotrafficking, were used in sort of underground new age medicine, if you will, but that they're, that's normally where the biotrafficking happens. When they're used authoritatively in hospitals, of course, they come from legal sources rather than illegal importation. Okay, so this made quite a splash in the news. And so what we wanted to try to understand was this, right? We knew that the leeches came from Russia, but we wanted to see if the study of iDNA could help us to pinpoint exactly where in Russia these species were from, or these specimens were from. And the second thing that we wanted to try to understand was if the leeches were collected from the wild or if they were from breeding stations. And we sort of assumed that if they were collected from the wild, then they would have a wide distribution of DNA inside of their bodies. Whereas if they were collected from breeding stations, then perhaps they would have, a smaller number and a specific number, maybe even only cow's blood inside of them. So what we did was we dissected about 240 of these leeches. We made a transverse cut across the crop in the back end of the body, the posterior, or basically the post-ceca, where a piece of the post-ceca were cut out. And we sequenced the DNA using 6 primer pairs, and they targeted 4 different loci that have been known to be able to separate between different vertebrate taxa. And we used parallel sequencing, shotgun sequencing, and a MiSeq platform. Basically, just one lane gave us enough information that we could pinpoint the precision, or with some precision, the collection locality for these species. So what we did then was we sequenced the DNA inside of the leeches, of course, using these 4 loci. And then we blasted those loci against different databases, mainly the NCBI NR database. And we used the blast comparisons, or the comparisons between sequences, to try to understand the identity of the DNA inside of the leeches. Now we used, I say rigorous blast identification here, because what we said was for a DNA sequence to be called a rigorous hit against the database, the identity of the DNA sequence needs to be 98% matching the target. And the coverage of the DNA also needs to be covered by 80% or more. And so basically, the top hit was deemed rigorous if it was 3% better than the second best hit. And by that, I mean, if it hit a specific cat, and then the second best hit was 3% below to another cat, then we said with some certainty that top hit was actually the identity of the DNA inside of the leech. Okay, so I'm going to move quickly into what we actually found. This is a table from the publication, showing a wide range of different animals that we found whose DNA was actually inside the leech. We found 16 different species inside of the leech. 11 of these actually had geographic localities attached to them as we move forward. I'll reiterate that when we get there, but out of these 16, 11 of them helped us to actually pinpoint the locality of where these leeches have been collected in Russia. Now, beyond those 16, we did get a hit against Felis caeus, which is the jungle cat. Not Felis catus, but Felis caeus. But actually, Felis caeus and Felis sylvestris and Felis catus are the genes that we use to separate these species are so similar that they cannot be used to separate these 3 species. Now, that could be because this is a very shallow split in time. It could be that these 3 species have split up in a very recent time, so they haven't had time to accumulate changes in their nucleotide chains. But we got the DNA from what we think is either Felis caeus, Felis sylvestris, or Felis catus. And the two contenders that rose to the top due to our bioinformatics or through bioinformatic techniques was Felis caeus and Felis sylvestris. Now, we couldn't really pinpoint which one we were getting, and this has very strong implications as to where we actually say that these leeches are from, as we'll see moving forward. So what I'll show you is maps that use Felis caeus, maps that use Felis sylvestris, and maps that use just Felis as it is, or without Felis. So I just wanted to mention that Felis caeus actually the jungle cat is quite restricted in its distribution, and that allowed us to pinpoint the distribution of these leeches. But there is question to this, right? We're not entirely sure that the DNA that we got out of the leech actually belongs to Felis caeus. Okay, so that's the question mark there. Okay, so what we did then was we compared the species that we found inside of the leech to different databases across the world, IUCN, GBIF, and FishBase, to try to find where the species that we found inside of the leech are distributed across the world. So what you're seeing here is a heat map of the 16 different species. The redder you get, the more of those species are actually present in that country. As I said, 11 out of the 16 species had detailed distribution data, so those were most heavily influencing the results. At the top here, you see the heat map without Felis caeus, which is the same as when we used Felis sylvestris. And B here you see with Felis caeus, and you see that there's no difference in the overall distribution of those of the different 16 different taxa there. Whereas you know, if we include Felis caeus or if we don't include Felis caeus. So I only wanted to show that to give you reason to believe me basically in this, that these regions are where the leeches were collected, and we'll get closer as we move along. Okay, so basically if we zoom in to those areas, the 16 different species that we found in the blood inside of the leeches allowed us to pinpoint the area to be somewhere in around the Black Sea, the Caspian Sea, the Eastern Mediterranean, as you see here in Figure A. If we move down to Figure B, and if we take these 16 different species that we have and where they exist in the world, and if we exclude Felis caeus, then we have a region around the Caspian Sea, the Northwest Caspian Sea, the Eastern Black Sea, and basically we're talking about a couple of different countries here, in particular Romania and Russia. And we know, of course, that these leeches came, at least were on the plane from Russia to Canada. And so by just excluding Romania from the rationale of the study, we can say that we probably have leeches that come from the northeastern part of the Caspian Sea. Okay, so in C here you see the very local distribution. If we do include Felis caeus into this study, if we believe that hit that we got for the DNA inside of the leech was actually from Felis caeus, then that allows us to pinpoint with a lot of precision actually exactly where the leeches are from, which seems to be the northern part of the Caspian Sea. Now, just to reiterate, Felis caeus is not a well-distributed species. It's not a well-known species, and because it only has been, you know, its distribution is quite narrow. That's why it was so important to us to try to test if we should include that species or not, right? Because when we included that species, it allowed us to narrow down the point of distribution or the point of collection of these leeches quite dramatically. As you can see, right?
Sorry, Sebastian.
Sure.
The Turkish translation is not gone.
Okay.
Can you manage it in the file site?
Sorry, the Turkish translation is not there.
Well, the problem is fixed, so you can continue.
şu an sorun giderildi. Teşekkür ederiz.
Thank you. The problem is fixed. We can go on.
Okay, perfect. Thank you, Dr. Ceylan. Okay, so between essentially we're looking at C and D here. So the top right and the bottom right. And this is the difference between using Felis caeus or instead using Felis sylvestris, which is a very much more widely distributed cat. And so basically, if we believe that the sequences come from Felis caeus, which is these two different species are within 2% range in their genetic variation, and so it's very difficult for us to say with certainty that we have sequenced Felis caeus or that we've sequenced Felis sylvestris. But if we believe Felis caeus, then the leeches were most likely collected from the northern part of the Caspian Sea. And if we believe that we have actually sequenced Felis sylvestris, then the western part of the Black Sea seems to be the region in which all of the species that we sequence within the leech co-occur in the same area. Now we know that area is Romania. We know that the leeches came from Russia, so we are inclined to believe that Felis caeus, or hope that we've sequenced Felis caeus there, and that the leeches were actually collected from the northern part of the Caspian Sea. Okay, so because of these 16 different species that we found, and because we found both birds and reptiles and amphibians and mammals inside of the leeches, we are inclined to believe, and I'd like to hear people's opinion on this, because you might know more about the leech breeding sites, especially in Europe, than I do. But we tend to think that these leeches were collected from the wild, given the amount of DNA inside of their bodies, but also the sort of species composition of the DNA inside of their guts. We believe that these are actually wild-caught leeches, but I'd love to discuss that with anyone who has seen the leech farms in this region of the world. Dr. Utevsky certainly, who I've spoken to about this as well, has some experience with these. So simply to conclude the discussion, leech iDNA has been used to find out the global distribution of mammals in particular, because they're charismatic and a lot of people care about them. They have been used to solve crimes, and now they've also been used to pinpoint the area of collection for contraband leeches. Now, this is the first time that iDNA from leeches has been used for this particular purpose. It also gave us a lot of insights into leech diets in general, and this is something that we continue studying here in my lab. Try to understand the leech diets, and this gave a lot of information about stuff that we didn't know already. It turns out that not only hemodipsid leeches, not only the terrestrial leeches in Southeast Asian tropical jungles can be used for iDNA purposes. Even freshwater species like Hirudo verbana, the species that we found in this particular study, can be used. And so this was the first time the freshwater species had actually been used for iDNA purposes. And of course, we understand that there's more utility in leech iDNA than simply determining mammal distribution, which is what a lot of studies have focused on. But it can tell us a lot about climate change. Of course, we've seen already that it can help in stopping crimes, or sort of helping in determining perpetrators of crimes. And of course, to tell us a lot about leech diets in general, which informs us on the behavior and ecology of the leeches. And again, I just wanted to mention all of these collaborators, which helped with the study, and so I'm the lucky one that gets to talk about it. But they were equally important to all aspects of this study. Thank you, everyone. If there are any questions, then I'd love to take them.
Thank you, Sebastian. Thank you for your report. Now we can see that leeches, medicinal leeches, can be used in forensic and to study biogeography and ecology of mammals. So actually, I expected that leeches, medicinal leeches, feed mostly on amphibians, but now
Yeah.
we can see that birds and mammals are important sources of food for medicinal leeches. So this is very new for us. Thank you very much for your contribution, and maybe some questions, but actually we are out of time, and I suggest that we go further and